Hello, students, and welcome to my video on genetics for Instructional Segment 4, Inheritance of Traits in the Living Earth NGSS curriculum from California. It's what we follow here at TASM. Just like all my other videos, this is not for profit, solely meant to help students. And let's rock and roll. All right, if you haven't watched the first video on DNA, I'd encourage you to watch that first. So here's a picture of Gregory Mendel. Camel on Masira Island, and a portrait of me from one of my students. Thank you. And how are we all tied together? We're all tied together through genetics, right? And so the big question in the DNA unit was, how does the structure of DNA lead to its function? And the big question here is the story of you. Why are you the way you are? Why do you have your blood type? Why do you have your certain traits such as skin color or eye color, etc.? And what are the genes at play there? And then hopefully you can tie it all together. You can see how different forms of genes give different proteins and how those can be selected for or against with evolution, our previous units. So let's jump in. This is fascinating stuff, and it's many students' favorite unit in biology. We got a little crunched for time at the end, so if you need some help, I've put the Amoeba Sisters videos for genetics right here underneath the Unit 4 assessment and classroom. I also have the CK12 textbook. I've been having you look at that for the warm-up problems in class, and I think it's high-quality material and worth checking out. You'll have this video and study guide as well posted right here, and I'll make a separate bullet point for my videos as well. Okay, and sorry, the standard is apply concepts of statistics and probability to explain the variation and distribution of expressed traits in a population. So different genes are going to give rise to different expressed traits, different alleles or forms of those genes, and then they can be acted upon by natural selection. All right, and so let's dive in and let's use an example. It's always easier to learn through examples. And so our phenomena for this one was we studied labor congenital amaurosis. And so we watched a video of Molly and of the genes as medicine, and how they tested it on basset hounds, and then they tried to help Molly with her eyesight. And so what was wrong with Molly? Well, Molly inherited a mutated RPE65 gene in her eye from each parent. Her mom and dad were fine because they have one dominant allele that made their eyesight okay, and then they had this recessive allele that carried the mutation. It's a rare mutation too. But Molly, unfortunately, inherited both recessive alleles from her parents. So she has this red protein here that is not able to help her photoreceptors work in her eye. I just love this video. I just think that this picture and the animation and video really helps tie it together for students. So you can see the passing down of traits. If she had just inherited this chromosome from mom, she would have been fine. Obviously, if mom and dad could have picked, we're always going to pick the best things for our children. They would have wanted to pass down the dominant allele or the non-mutated one, but we don't get to choose. It's not in the cards right now. And so what we do is we make sperm or eggs, and it's random which allele is passed down. It's called the principle of segregation from Mendel. And so this is going to be on an autosome, and it's going to be an autosomal recessive disease, and we will go through that with the pedigrees and learn more about it. So just remember to tie it back to this phenomenon, and you can think about it. And I think most of you all really liked that video and liked learning about Molly and the happy ending that was there for her. Here are a picture of your human chromosomes. Now, we tend to think just about humans in genetics, but all organisms do genetics as well. And so here for humans, we have 23 pairs of chromosomes. Numbers 1 through 22 are called autosomes. And notice how they're identical. You're going to have one from mom and one from dad, and they have the same genes on them. Number 2 from mom and number 2 from dad. Number 3 from mom, number 3 from dad. Number 4 from mom and number 4 from dad. They're the same genes, but they may have different versions of those genes or different forms of them called alleles. So maybe mom gives you the allele for blue eyes and dad gives you the allele for brown eyes. And as we went over in class, brown eyes would be dominant from the OCA2 gene. But then there's other things at play there. There's more than one gene for eye color, which is why we have a vast diversity. I believe there's 16 of them that contribute as our latest understanding. What's fascinating here is if you go to the sex chromosomes, you'll see XX is female and XY is male. You see that the X is much larger and expresses more genes than the Y does. Men will be more likely to inherit sex-linked diseases. So can you describe Mendel's experiments and what he figured out? So here he bred a tall and a short pea plant. They all came out tall. And then he bred these F1. This would be the P generation, the pure breeding generation. This would be the F1 generation. He bred this generation again, and you got 3 out of 4 tall and 1 short. That's on average, right? It's not like every time. Like, think about it. On average, it's 1 boy, 1 girl. But you might have a family of 2 girls. You might have a family of 2 boys. It's on average. And so this is the P generation, F1, F2. So what's going on here? Well, it's pretty cool, right? That these short alleles stuck around. That's the big part to get here. The short allele wasn't gone. It wasn't gone forever from the population. The plant didn't come out medium. It disproves blending, and it showed that alleles can be passed down through generations. If it was a blending model, then tall and short would have made medium, which would have made medium. It's very hard as humans for us to get away from this blending model because we do look like a blend of our parents, and we might be a blend of their heights, or we might be taller than them, etc. The problem for humans is that we have many, many, many genes contributing to one phenotype or one physical trait, such as skin color. There's a lot of genes that play there. So there's a lot of different Punnett squares that are being completed inside of our body to give us our skin color. So let's just keep it simple and think about Mendel, and then we'll get more complicated later on. So he did this with pea plants, and he kept them separated, and he was able to breed who he wanted to breed with who. And so it was a very effective way to figure this out. Here is just a little more terminology for you. This would be a homologous pair of chromosomes. Homo means the same, so same genes. But remember, they may have different forms of the genes or different alleles. The locus is the place of the gene on the chromosome. And so we've mapped chromosomes, and we know where their genes are, etc. And so here's a good example. Here you could have 2 chromosomes. This is one from mom and one from dad. This would be a 1 homologous pair, 2 chromosomes, 3 genes, and 6 alleles. So stop the video there and see if you can construct some knowledge with that. We would have 46 chromosomes for us, 23 homologous pairs, about 30,000 genes, and then lots of alleles. Notice how for this gene here, the light blue one, the individual will be heterozygous. Hetero means different. So we're going to have 1 dominant allele and 1 recessive allele. Heterozygous as well here. 1 dominant allele, 1 recessive allele. Homozygous, dominant here. Homo means the same. So 1 big G, 1 big G. So the 2 are the same. Or they could have been homozygous recessive. 1 small g and 1 small g. Here is another picture of Mendels Experiments. So violet and white flowers, so purple and white. If you breed them together, they all come out purple. Remember, P for parental generation or pure breeding generation. And then here is the F1. So these are all going to be, if you think about it, if we use capital P to represent the dominant allele, these ones are all going to be capital P, because they always come out purple. These ones are going to be small p, because they always come out white. And these ones, because it's the breeding of these 2, the purple and the white from the P generation, are all going to be heterozygous. Small, big P, small p. Then if you breed these 2 together, you get your classic 3-to-1 ratio. We're going to have 3/4 purple and 1/4 white. Let's see why. All right, let's learn about a Punnett square. There's got to be a faster way to erase. I'll figure it out, Screencastify. I'm coming for you. Here are the traits that he looked at and figured out all this information with. Here is a Punnett square. And so here would be the classic one we were just talking about, yellow and green, pure breeding yellow, pure breeding green. And we went over this in class. The principle of segregation says that only 1 allele goes into each gamete and that it's random. So here, 1 big Y goes into the sperm or egg, one little y goes into the sperm array. You then combine those together and you get the F1 generation. This F1 generation is going to be a big Y and a little y. And they're all going to come out yellow because the big Y is dominant. We are now going to breed yellow by yellow. So we're going to have a big Y, little y, big Y, little y. This is once again the principle of segregation. We're going to have one allele into each sperm array. So if this plant right here is making sperm, half of the sperm will have the big Y and half of the sperm will have the big or the little y, right? Or let's say there's eggs. Half of the eggs would have the big Y, half of the eggs would have the little y. The outsides of a Punnett square are the sex cells. They are the gametes. They only have one allele for each gene. The inside of the Punnett square is going to have two alleles for each gene because it's going to have the homologous pairs of chromosomes. So here, sex cells, it only has one copy of each gene, one allele of each gene. Here on the inside, it's going to have two copies of each gene or two alleles. So when you cross them, you're going to get yellow. This would be homozygous dominant. Here, you're going to get a big Y and a little y, so you're going to get a heterozygous. Here, a big Y and a little y, so another heterozygous. And here, little y, so a homozygous recessive. All right, and so it's going to get a 3/4 yellow, 1/4 green. You can think of each of these boxes as 25%, right? And it should add up to 100. So 25% chance yellow, 25% yellow, 25% yellow, 25% green. So if you were to have 1,000 flowers, you would say 75% of them would be yellow, or around 750. Green would be around 250, right? Or 25%. And you can do some math to see if it's following that. It's called a chi-square test. All right, a test cross. So let's imagine that you had this yellow seed, but you don't know if it's from the pure breeding generation or if it's from the F1 generation. This is important to you because you're about to plant a huge field of them and you want all yellow flowers. You can do a test cross. And so you take a couple of those yellow seeds from that stock and you would breed them with the green seed right here. You don't know what the second allele is here. You know it's a big Y because it's yellow. It has at least one dominant. It's either big Y, or big Y, little y. And so if we breed it with the green here and they all come out yellow, then we have to use logic to know that this allele was a big Y. Because otherwise, they would have come out half green. Since it was a big Y, this one was all the seeds came out heterozygous. Big Y, little y, big Y, little y, big Y, little y, big Y, little y. Now, if they'd come out half green, then you would know that this yellow seed was heterozygous. It has a little y here because half of them came out green. And I gave you that little y, little y, little y. This is called a test cross. This is going to be really important later on if you take AP bio. We're trying to figure out if genes are linked. And so we will test them with a homozygous recessive and see what's going on. All right, try this problem. Okay, can you set up this Punnett square? Hopefully you got this, that it's going to be 3/4 purple and 1/4 white. Here is an animation just showing you from the amoeba sisters for hairy and hairless guinea pigs. Hairy is going to be dominant. Hairless is going to be recessive. Pretty cool, huh? So you can watch that again. They bring, so now this one's going to be a heterozygous by homozygous recessive. Awesome. Thank you, amoeba sisters. Excellent. They got really good videos. I really like them. Here's going to be a homozygous dominant with a homozygous recessive. So here, this is that P generation, right? These are P generations. So that's giving you an F1. Okay. So to do the Punnett square, you bring them over and down. Normally, we write the dominant letter first because it's just easier for us to read as humans. All right. You most likely did that in middle school. So now we're doing dihybrids, and this is 2 traits at once, right? And you're really 30,000 traits at once. And so for a dihybrid, we're going to start with, and I did this in class with you. This is the classic heterozygous one. So we're going to have a big Y, big R, little r in the F1 generation. We're going to breed them together. But first of all, we need to figure out all the possible gametes, all the possible sex cells. Remember that a sex cell only has one allele of each gene. So we're going to use FOIL. And here it is. We're going to take first, so big Y, big R, first, outer, big Y, little r, right here, inner, little y, big R, and last, little y, little r. So they did it in a slightly different order, but it doesn't matter which order you put it in. But the FOIL will help us to get all those possible gametes. Here's the important thing, students, as you're looking at this. Notice how these only have one version of each gene. They either have one Y or one R, and it could be a big Y or a little y, a big R or a little r. I saw some students struggle with this on their jam boards, and we went over it a couple times to hopefully clarify that. Once you cross this, if you cross two organisms that are heterozygous for both traits, you're going to end up getting a 9 to 3 to 3 to 1 ratio. And this is called a Mendelian ratio for the double heterozygotes. So you get 9 yellow round, 3 green round, 3 yellow wrinkled, and 1 green wrinkled P here. So go back through your Jamboard, go back through your notes, and check them out before you write them out. Here comes our FOIL. Thank you, Amoeba Sisters. First outside, inner, and last. So I'm going to let that play. You all should try it. Thank you. All right. What if we don't get Mendelian ratios, such as freckles and red hair? They're often seen together, more than a 9 to 3 to 3 to 1 ratio. That means that they're on the same chromosome. And this is assuming what we call independent assortment. And by independent assortment, it means that Y's and R's are passed down independently of each other. They don't, it doesn't matter what one is doing for the other. For example, when you go home at night, it doesn't matter what one student in the blue group is doing versus the red group. I mean, you might hang out online, etc., but you independently do your homework, right? And then you might come back together in class and share your results. So it's not like the R is linked to the Y in this example. But we know that you only have 23 chromosomes and you have 30,000 genes. So there have to be multiple genes on the same chromosome. Genes that are on the same chromosome can be linked together and will violate Mendel's principle of independent assortment. And when that happens, they tend to be inherited more often together. All right, practice this dihybrid, okay? And so read through it. You'll need to assign some letters for the alleles. Here's what you could have gotten, right? And so if you cross it out, this is reviewing the product rule. And so we went over this in our Jamboard. And so you would be, whatever you're looking for, you do the first Punnett Square with the same genes. And so heterozygous, big B, little b, big B, little b. You do this here. And so let's say that we wanted to do the probability of a yellow, let's say smooth-eared dog, right? Yellow would be 1 out of 4. Smooth-eared would be 3 over 4. So 1 over 4 times 3 over 4 would be 3 over 16. And so if you do the dihybrid, it would come out like that, which is pretty cool. This one said, what fraction of F2 puppies have yellow straight fur? So, no, that's exactly what we just did. All right, good. There we go. All right, so you can review those. You can check out the work there. You can also check out the Amoeba Sisters video. The product rule works with multiplying, right? It has to be, they have to be together. There's the sum rule, but we didn't go over it this year dealing with OR, and so it's just something we didn't get to this year. So for our purposes, we'll only do the product rule. All right, what are some exceptions to Mendel, right? Some teachers argue to not even teach Mendelian genetics because there's so many exceptions to it. I think it's helpful to have a basis for it. And so there is incomplete dominance, and that's when neither allele is going to be dominant over the other one. So here inside the cell, if it has a big R and a little r in the metabolic pathway or in the biochemical pathway to make this pigment, it's going to come out pink. So this looks like blending, right? But if you were to cross two pinks together, you then get the red and the whites and they come back out. And so this disproves blending, right? And this is the snapdragon plant here because the reds and the whites can come back out, which is pretty cool. So this is just showing that occasionally there won't be dominance. All the examples we've dealt with so far, the big R was dominant over the little r. But if I tell you in the problem that there's incomplete dominance, then it will be in between the dominant and recessive phenotype. So here it was pink in between these two. The way I remember this is if you turn in something incomplete, nobody wins. All right, codominance is like a couple, right? And each person should be expressed. And here is a good example of a Rowan cattle. So they're going to have both red and white fur expressed. And so here will be RR and here's WW. So RW is going to have both the red and the white expressed. You can also see this with blood types. For example, A and B is what we went over in class. Here are your blood types. And so disregard the chinchilla thing here. We didn't get to that this year. So here would be A. So A is going to have these markers that make it an A. B is going to have these markers that make it a B. A, B will have both because they are codominant. So they're expressed together like a couple. Whereas group O will have none of these markers. They will have an absence of them. These markers can trigger an immune response. So I am B positive. So if I get a transfusion of A blood, my body will reject it because it will see these round ones instead of these square-like spikes. And it will say, hold on, John, this is different. This is not normal for us. This is non-self. And so we're going to send out an immune response to get rid of these cells. So it's important that you get transfused with blood that matches yours. Or you could get O negative blood because O doesn't have any of these markers on it, so it won't trigger an immune response. The plus part is the rhesus factor. And so here are the individual alleles. And so I went over these in class. The I stands for the presence of the antigen on the red blood cell. And then the antigen is this thing, this marker, the square or the circle. It's called a glycoprotein. Here's a cool problem set with it as well from University of Arizona. And so here is type O. Type O is not going to have that marker, right? And so AB is showing that codominance. You can be heterozygous or homozygous for type A or B blood. And that's pretty cool. So two heterozygous Bs, if you were to cross them together, could have an O baby. All right, so can you practice this? Mom is type O, dad is type AB. What can the kids be? Well, hopefully you get this. And we did a couple of these in class. Mom is type B heterozygous, dad is type AB. What can you get? Here's the heterozygous mom. Here's dad AB, so you can get A, B, or AB, but no O's. How about type B heterozygous, type A heterozygous? This is pretty cool, right? You can get A, you can get B, you can get O, you can get AB, you can get everything. My goodness, pretty cool. All right, sex-linked genes. Sex-linked genes are going to be on the X chromosome. And so we went over this a bit with hemophilia and with X-linked SCID, and we'll go over it more with Duchenne muscular dystrophy. This affects boys more often because they only have one X chromosome. So this is the most classic case where the mother is a carrier. So she's heterozygous for this trait on her X chromosome. So this tends to mess students up a little bit because you now have this sex represented with the X's and the Y's, and then you have the allele of the gene as like a superscript. So you got to practice writing these. So X, big H, X, big H, X, big H, X, little h. Remember, the sides of the Punnett Square are sex cells, gametes. So these would be eggs, and here are the sperm from dad. For a son, the son's going to get the Y chromosome from dad. The Y does not have the hemophilia gene on it. It doesn't have this blood clotting gene on it. So therefore, whatever mom gives him is what he's going to be. He's going to be, you could think about him as being homozygous for the dominant allele, or here homozygous for the recessive allele. And so the daughters, none of them will have hemophilia because dad's going to give each daughter the dominant allele, And even if they get the recessive one from mom, the dominant allele will mask the recessive allele here. But the sons could get it. And so you can do some fun genetic probabilities here. It's not fun if you have hemophilia, right? We do have medicines for it now. And we'll review how it worked in the royal family as well. It's quite fascinating. There's just so much with genetics. I could spend the rest of the year on genetics, but we got to get the move on to get the groove on. And so here you would say that 25% chance of a child with hemophilia. But if you know that it's a boy, then it's going to be a 50% chance that they have hemophilia. Here, let's look at some flies. And so here would be a red-eyed female and a white-eyed male. If red is dominant to white, can you complete the cross? Well, hopefully you got that they are all red-eyed, and that's pretty cool. But now we're going to have some females who are going to be heterozygous for this eye color trait, right? They're going to have the red and the white allele. So if we breed it again, then we should get something different. So let's do it here. Let's take the heterozygous red-eyed female with the red-eyed male. And if we breed them, we get, okay, they're still, all girls are still red-eyed, but half of the boys are white-eyed. This is the exact same as that hemophilia Punnett Square we just did. And this is what Thomas Hunt Morgan and Alfred Sturtevant looked at and figured out these ratios. Okay, so calico cats are pretty cool. And so we just adopted a calico cat, the one that was around the front doors of Tazem. And so the gene for coat color is going to be on the X chromosome. And it can either be orange or black. But females inactivate one of their X chromosomes early on because they don't want, you only need the proteins expressed from one of them. And so that activation is random. It's called a bar body, and the making of it is called lionization after Mary Lyon, the PhD geneticist who figured this out. And so we actually named our kitty cat Lion after Mary Lyon. And so there we go. The cat kept me up last night. It was really annoying, but it's okay. My kids love it. So that was too quick, but the class is coming up. So you can research a bit about calico cats and why they're most likely female and expressing this. There's a really cool gizmo on it as well that we just ran out of time that we can do with meiosis. And maybe we will later on in a year if we have some time. All right, here is the last bit, and this is on pedigrees. I sensed that it was a little frustrating for some of you all to try and figure out the genetic mode of transmission. Some worksheets that I've seen on the internet, et cetera, kind of give you what's going on and I just want you to interpret it. And I kind of made it harder for you. I was like, try and figure out what's going on in this pedigree and really challenge some of you all. And I hope you enjoyed that challenge. And so here are some steps. When you're looking at a pedigree, you're trying to figure out how the trait is passed on. You might not have enough information to figure it out. You might just have to say, we can't, with the amount of information we have available, we can't figure it out. You might be able to look at it and say, this is how this trait is being passed on. The trait could be a disease, or the trait could be something like eye color or dimples or something like that. The point of this is, if it's a disease, you could be a genetic counselor and you could talk to someone and be like, hey, you have a 25% chance of having a child with Leber congenital amaurosis. And so for all the classes, except for FBOC, we haven't gotten to do it yet if you're watching this video early, I had a little ethics thing where we talked for 30 minutes about, well, if you know that you're carrying a disease, should you have children? And like, what are some of the ethics that go into that? And it was a really fascinating discussion, and you got to do it in small groups, and you came up with some really interesting points. For me, I'm a carrier for Tay-Sachs. That's a lethal disease. And so my wife was screened and she wasn't. And so we knew that we could try and have children and they'd be okay and not have Tay-Sachs. So here, if we look at this trait, we see that it skips a generation. You need to look within the family lines. So here is a family line. Here is a family line. Here is a family line. Notice how in this family line, it skips generation 2. In this family line, it skips generation 2 as well. If it skips a generation, that is a recessive trait. All right, your next goal is to figure out, well, is that recessive trait autosomal on chromosomes numbers 1 through 22, or is it sex-linked? Remember that if it's sex-linked, it's going to be more likely to affect boys. So if you see a lot more boys affected, then it might be sex-linked. But you would have to do some logic here. And this is the fun part, right? And so you can look at moms and sons and dads and daughters. So this mom, if it's sex-linked, will pass it down to all of her sons, right? Because the sons are going to get a Y from dad, and you get an X from mom. So if a mom has an X-linked disease, all of her sons must inherit it. So far, it works here in this part, but it doesn't work here. Mom has a disease and the son does not have it in 2. So in 1-1 and in 2-2, mom has it, son doesn't. So it can't be sex-linked, right? It's not following that pattern. Here you can see that this daughter has it, but her dad doesn't. So it can't be sex-linked as well here. So if a daughter has it, her dad has to have it. Think about why. Dad has a Y and an X. He's going to give that one X to his daughter. So therefore, if a daughter has a homozygous recessive disease on the X, dad had to have it too. So this is going to be autosomal recessive. Let's look at some other ones. These are real pedigrees from our phenomena with Leber congenital amaurosis. You can see that it skips a generation, so it's going to be recessive. And then you can see that this daughter has it and her dad doesn't. So it's going to be autosomal. And that is the way that it's passed down, autosomal recessive. Once again, you're using those claim, evidence, reasoning skills with these pedigrees. If you talk it out, I'm going to give you points on the test, even if you get it wrong. Like if you're trying to give me reasoning here that shows that you're understanding what's going on. All right, I am lagging a bit with the internet here today. So we're almost done. X-linked recessive. This one follows all the rules. It can be X-linked recessive. Notice how a daughter has it, dad has it. There are no moms and sons with it. It has more boys than girls. Here's the trick. You can tell me, you say, Mr. Gibney, this is most likely X-linked recessive. And I can say to you, well, it could still be autosomal recessive. Right? You can't completely rule out autosomal recessive if you were to say little a, little a, little a, little a. It could be passed down that way. So you would say that the evidence points towards X-linked, but you can never rule out autosomal recessive. It's only the other way around, where you can rule out X-linked, but you can't rule out autosomal recessive. And then here's mitochondrial. Oh no, this should be on that one right there, the colored in. But the way this works is that if there's a mutation in the mitochondria is in the egg. And your mom is going to pass down mitochondria to you. Your dad only passes into the egg 23 chromosomes. And so all the other stuff, the mitochondria, the Golgi apparatus, the transcription factors, the proteins that are in the egg come from your mother. So slightly more than half of your genetic information is actually from mom. So if there's a mitochondrial disease, and the reason I included this is because we've studied the mitochondria in depth in Unit 2 in photosynthesis and cellular respiration. It affects all of her children. So if it's mitochondrial, it affects all of her children. And then only her daughters pass it on. So mom, all of her children, only the girls pass it on. Daughter passes it on, daughter passes it on. Right? This square should be right there. Sorry. So that's a mitochondrial mutation, and you could look up one called Leigh syndrome to learn a bit more about it. Here's, this is a really tough one. And it's a tough one because students think, oh, is it skipping generations? It doesn't. It doesn't skip generations. And this is actually going to be autosomal dominant. And here's how I can tell. Individuals 2-5 and 2-6 have children. If it was recessive, all their kids would have to have the trait. But because one of their children don't, then it's a dominant trait. Because they would have to be heterozygous. They would be big D, little d, big D, little d. And this child right here, 3-9, would be little d, little d. It's the only way it works. So if you see two individuals affected and they have a child that is not affected, then it will be autosomal dominant. Right? And so here's your bonus research with other cool things to check out. There's one thing to note in pedigrees, and I said this in class, but there can be instances where a disease just arises in a family. And that can be due to mutations during the creation of sperm and egg. And so, for example, there can be a mutation in creating a sperm or egg. And if that mutation happens inside of a gene, or if it happens with a promoter with a gene, it could start a genetic disease in a family. There's so much more. Check these words out. Try and learn about them. It's just absolutely fascinating. I hope you enjoyed. Take care and learn some more genetics.